Good afternoon, everyone. Welcome to this webinar entitled Quantifying Medical Ultrasound, an Introduction, presented by Dr. Andrew Hurrell. The AIUM is accredited by the ACCME. This webinar is designated for one AMA PRA Category 1 credit. By the conclusion of this webinar, participants should have an overview of why we need ultrasonic metrology, be able to discuss the various parameters we may want to measure and what tools we may use for that purpose, and understand that all sensors are imperfect, that measurement accuracy arises from the ability to recognize, quantify, and correct the limitations of measurement tools. As an ACCME accredited provider, it is the AIUM's policy to ensure that the contents and quality of this activity are balanced, independent, objective, and scientifically rigorous. All individuals who are in a position to control the content of an educational activity are required to complete a disclosure form. All disclosures are reviewed by the AIUM, and any conflicts of interest are resolved or managed prior to an activity. Dr. Andrew Hurrell and the AIUM staff members have no disclosures. At the conclusion of the webinar, you will be able to access the CME test and evaluation located on the AIUM website. Participants who complete the post-test with a grade of 70% or higher will be awarded one CME credit. Credits for this webinar are approved for physicians, sonographers, and radiologic technologists. During today's presentation, if you have questions for the presenter, you may type them into the question box on the right side of your screen. You will be able to submit questions throughout this webinar, but the presenter will not begin answering until the end of the presentation, at which time he will answer as many questions as possible. A recording of this webinar will be available on the AIUM website. And now, we're pleased to present Dr. Andrew Hurrell.
Okay, well, depending on where you are in the world, good morning, good afternoon, or good evening. Thank you to Darcy. As we can see from the title screen here, we're going to be talking about the quantification of medical ultrasound, and this is an introductory webinar. Just to let you know that we may be looking at some images of some precision acoustics products whilst we're here, and I am co-owner and employee of that particular company. So it's first to look at the introduction here, that this is the first introductory talk in a series on ultrasound measurements. Thereafter, Jim Gessert from Asatara will be talking about issues relating to diagnostic ultrasound, and Sam Howard from Onda will be discussing the measurement challenges associated with therapeutic ultrasound. I'd also like to begin by discussing what are the aims of this particular presentation. Firstly, why do we need to quantify ultrasound? Who regulates and standardizes these measurements? Which parameters should be reported? What measurement tools are available? And how can we ensure that the measurements we make are the best ones possible? So to address the first of those questions, why do we need to quantify ultrasound? We probably ought to step back and take a look more globally at ultrasound generally and realize that ultrasound therapies are in common clinical practice. We'll look at an example where some mechanical bioeffects are clearly evident, and we'll do much the same for thermal bioeffects. We can ask the question, are there any thresholds that we need to observe? And we'll certainly then discuss, can we report our output well enough to ensure that subsequent studies that may be following on from any particular work that we're involved with are repeatable. So let's start with the first of those. I'm going to start talking about the practice the technique of lithotripsy. Lithotripsy is where we're using highly focused ultrasounds to break stones. And an example of one of the Creative Commons images here is of not a typical stone. In this case, it's a rather rough structure, but there are all sorts of variations one can find in stone morphology. And the lithotripsy itself is likely to be the sort of thing that we would see actually on the image on the right-hand side, where we've got the shock head down here next to the bench on which we're likely to find the patient lying. Now, in this particular methodology within lithotripsy, we find that as those highly concentrated shock waves are focused down upon the stone that we're looking to treat, a number of effects could happen. I'll go through what those effects are. We can start off with cavitation. This is where we have bubbles collapsing immediately in the presence of the stone, and these can cause high-speed jetting, which damages the stone. Sporulation, this is where we can get bits of the stone flying off its rear surface. We could find that there were shear stresses induced in the stone and also quasi-static squeezing. All of these mechanisms and potentially more can go to contribute to stone breakup. So clearly there's a range of mechanical bioeffects there. We'll also now start to talk about thermal bioeffects. What I've got here is a simulation of a focused ultrasound beam. Now, if we look at this, as we see where the focal region, which is where we've got this black and white cigar-shaped region that I'm highlighting with the mouse, is shown. This is the area where we've got the highest concentration of ultrasound, and you can see as all of those waves converge upon that point, there's a very obvious lozenge point. Now, this is a simulation, and one may ask, well, how does this translate into clinical practice? So if we now look, I'll wait for that simulation to restart again. If we now look particularly at that cigar-shaped region, this is an example of a lesion that was formed in liver tissue, and this was recorded by Gail Tahar at the Imperial Cancer Research Institute at the Royal Marsden Hospital. And you can see that, again, you've got this very good correlation in shape between the simulation and the histological slice that we see there. And in this case, the dominant mechanism is heating. So clearly then we can ask the question, just how much ultrasound do we need? In a therapeutic situation, we're looking to cause deliberate bioeffects. However, if what we're looking at is a diagnostic situation, then we're trying to minimize bioeffects. And at that point, there needs to be realized that there's a threshold somewhere between the two. And depending on whether we're looking to undertake a therapy or whether we're just looking to diagnose something from the tissue, we can set some kind of threshold level. Exactly what that threshold level and where it is often not the responsibility of the experimenter, but we'll go on to talk about who may be looking at the regulations in that area. So we ask the next of our questions, who regulates and standardizes ultrasound measurement? And this is generally accomplished via three different groups. We have the regulatory authorities, the sources of standardized procedures, and then auditors or notified bodies or scientific certification organizations. The role of regulatory authorities is to establish what needs to be measured and reported. So clearly in the US, we have the Food and Drug Administration. A little further north of the border, we've got Health Canada. Across the Pacific, we've got PMDA in Japan. In my own country, we have the Medicines and Healthcare Products Regulatory Agency, and they operate under the same set of regulations as are specified by the European Union in the regulations that come out of the Council of Europe. Who recommends how quantities are measured? Well, the biggest source of ultrasonic related regulations at the moment is the IEC, the International Electrotechnical Commission. This is formed with representatives from many nations around the world, and there's active participation from at least 40 nations in the ultrasound group. There are also local US regulations, particularly UD2 and UD3, which people may be familiar with, that have come from AIUM and NEMA. And it's also worth bearing in mind that because of the harmonisation work that's ongoing within the IEC, many of the stipulations of UD2 are now making their way into harmonised regulations that apply worldwide from within the IEC framework. And then also the ISO organisation, International Standards Organisation, will have recommendations in terms of how we may be undertaking good laboratory practice. We now also need to look and see whether the implementation of those regulations is done appropriately. And this is where we may have the role of auditors or notified bodies who will be assessing whether equipment manufacturers and their devices satisfy legislation. You may well be familiar with a number of these names, including underwriters' laboratories or members of the TÜV group, British Standards in Industry, or Lloyd's Registered QA. All of these undertake a similar function. The next of our major questions was which parameters should be reported. And before we do that, we need to start to break that down into a few smaller sections. So we'll start to consider the difference between directly measured and derived quantities, the difference between peak and average quantities, and specifically in the context of ultrasonic intensity, we'll ask ourselves which one. So let's start by looking at the difference between some of the directly measured and some of the derived quantities. The quantity which is most commonly measured directly is ultrasonic pressure. We'll talk about devices that we might be able to use for that particular purpose a little later on. Ultrasonic power is an interesting one. We can measure it directly with a radiation force balance, but we can also measure it indirectly with a hydrophone. We can also measure beam width and or focal length directly, and we may also be able to measure issues such as the frequency, the pulse repetition rate, and the pulse or burst duration. In terms of derived quantities, ultrasonic intensity is the probably most principal ultrasonic derived quantity, and this is normally derived from an ultrasonic pressure waveform. And we then need to make some calculations on that in order to get to a point where we can reach ultrasonic intensity. As mentioned, ultrasonic power is also something which we could measure with scanned hydrophone. It's actually a value that is derived from the already derived ultrasonic intensity, so it's a very indirect measurement. And finally, for those of you familiar with the output indices, the mechanical and thermal indices, these are again derived quantities. We're making measurements of a certain parameter and then we're undertaking some calculations on those. Now let's take a look at the difference between peak and average quantities. What I'm showing on screen here is a reasonably typical diagnostic ultrasound waveform. And it's relatively easy to determine where the peak of that is. In fact, what I'm showing there is the peak positive value. Notice that the peak negative value is somewhat different from that. And I'm sure this is something which, when Jim Gessert talks about diagnostic ultrasound, that will feature in his presentation. So clearly, given that we've got a peak value, that's not necessarily representative of what's going on in the rest of the pulse. And in fact, if we start to look at what might be the pulse average value, we can see we get something much lower. And I've tried to provide an indication of the typical pulse duration. It's worth noticing that in most of the standards, because of the way that pulse duration is calculated, it's not exactly from the zero crossing point on the beginning of the trace to the zero crossing point at the end of the trace, but is often determined in terms of a quantity that relates to the energy in the pulse. And in this case, the pulse duration is giving us a value that's about half a microsecond. But it's also worth bearing in mind that for many ultrasonic situations, the pulsing regime is far from continuous. In fact, you might find that pulses only repeat themselves once every 10 microseconds. Well, given that we've only got about half a microsecond pulse duration, we can see that the time averaged value, so average over all time rather than just the duration of the pulse, is going to be lower again. And the arguments that apply with regards to, in this case, a temporal variation are equally valid for spatial variations as well. Are we interested in just the biggest single value somewhere in the field, or are we interested in the average over an area within an acoustic field that we're looking to quantify? And it's exactly these kind of discussions which then lead us on to thoughts about intensity. We often hear the phrase ultrasonic intensity quoted, but actually it's quite important to start considering which ultrasonic intensity. So if we consider now, we have got spatial peak and spatial average intensities. We've already considered graphically the fact that we've got temporal peak, temporal average, and also pulse average intensities. But there's a further complication as well. A lot of our measurements are made in water. Water has very little absorption, certainly at frequencies below about 30-40 megahertz. Whereas if we're trying to work out what's likely to be going on within tissue, then we need to make a correction to our in-water values to produce what is more likely to be found in a tissue medium. And therefore we can have any combination of, say for example, spatial peak, temporal average, attenuated tissue correction, or spatial average, temporal average, linearly measured. So you can see we have quite a combination of intensities already. And it's important to realize that these can be very different in value. The spatial peak, temporal peak value could be vastly different to the spatial average, temporal average value. And we could be talking orders of magnitude difference here. So whenever we are reporting an ultrasonic intensity, we need to be careful which one are we declaring. So we'll now go on to look and see what measurement tools are available for someone undertaking measurements. We have hydrophones. Now, a hydrophone is a device that measures pressure, but it only measures in one location. And therefore, if we have a complex field, we may need to move that hydrophone around to map the field. And this often would necessitate some form of hydrophone positioning system. We also have radiation force balances. Now, these are devices that measure power directly, but they only measure the whole beam. There's no opportunity for spot measurements with a radiation force balance. I'm now going to go on and talk about those two different measurement tools in a little more detail. Firstly, we can ask the question, what is a hydrophone? So what I've got here is a hydrophone element. In this case, I'm assuming this to be some form of piezoelectric element. There are other acousto-optic sensing mechanisms, but for the sake of simplicity, I'm going to concentrate on piezoelectric devices. If we have a source of ultrasound, then as that arrives at the hydrophone element, it will cause that element to expand and compress. And as it does so, we get a varying voltage produced on our voltmeter. That voltmeter is probably going to be something like an oscilloscope, which allows us to record a waveform, and then we can digitally capture that for post-processing later. And here we can see we have our varying voltage in response to the movement of the hydrophone element. We need to bear in mind this is a direct measurement of ultrasonic pressure, or at least a voltage that is proportional to ultrasonic pressure. How we get absolutely to ultrasonic pressure will require our device to be calibrated, and we will talk about calibration in a little while. So let's start talking about the different types of hydrophones. There are a number of commonly available types, and I'm going to give you a few examples of each. This is not an exclusive list, it's just to give you an idea of the different devices available. So we're going to start off with some needle hydrophones. As their name suggests, they look a little like a sharp needle. We can find them available in a range of active sizes, from millimeters across to tens of microns across. They're good general purpose measurement devices, commonly used, and have got a relatively small scattering cross section. So if we're looking about or worried about the signal that may be reflected back towards our transducer, these are often a good choice of device. However, as with all probe type hydrophones, the very tip of the sensor, which I'm highlighting at the moment, which is where the active element is, can also be a source of diffraction. And this diffraction can limit the sensitivity of these devices, particularly in terms of how they respond as a variation of frequency. The next device that we'll look at is often called the golden lipstick hydrophone. These hydrophones are deliberately designed to try and deal with some of the limitations that one finds with needle hydrophones. And thank you to Petra Yam from Onda Corporation for the provision of this image. We also have fiber optic hydrophones. Now these tend to be very small indeed. Typically we have exposed glass substrates that are of the order of about 100 microns across, and the sensing element here can be only tens of microns or maybe even just 10 microns across. And then finally we have a membrane hydrophone. Membrane hydrophones are physically much larger than some of the needle hydrophones. In fact, any of the probe or semi-probe type hydrophones that we have seen in the other images. They have a very broad band response that covers a very large frequency range, but because they are a large membrane, this can be set into resonance by low frequency waveforms or continuous waveforms. So one has to be a little bit careful about using these in continuous wave fields that we may discuss a bit later. So this is a range of four of the more commonly available types of hydrophone. Now let's go on to look and see what is a radiation force balance. Typically we have an accurate microbalance. As drawn at the moment, this has got a tank of water above the microbalance, and we also have, in this case, an absorbing target within there. Some devices have got targets suspended underneath the microbalance. Some of them have got reflecting rather than absorbing targets. There are a range of different options available. So again, when we start to look at our source of ultrasound, we can see that we see a displacement of that top pan balance due to the interaction, the pressure of the ultrasound on that target. And that allows us the capability of directly measuring ultrasonic power. The big advantage of this method is that the conversion factor between ultrasonic power and the mass that we may measure on our balance can be determined purely by two constants. It's affected by the acceleration due to gravity and the speed of sound in water. So we can get a direct measurement of ultrasonic power that's not dependent upon a number of other factors. However, the disadvantage of this method is that the conversion factor that arises from those two constants means that we have a sensitivity that is of the order of about 67 to 70 milligrams per watt of ultrasound. So if we're looking to measure typical diagnostic levels of ultrasonic power in the tens of milliwatts or less, we need very, very accurate microbalances. So we need to ask ourselves, how can we make the best possible measurement? And this is a multi-stage process. We have to start by identifying the limitations in whatever it is that we're looking to do. Having identified those limitations, we may want to try and quantify them, and if they're a limitation of the equipment itself, we may want to try and calibrate for any variations that we see. Having calibrated our equipment and identified limitations potentially in our experimental method, we can either use that calibration data to correct our measured data to get a more accurate value, and maybe correct our experimental method to allow us to counter some of the limitations that we've already identified. And then at the end of that process, we can come along and evaluate our uncertainty. Now, it's important to realize that all measurement tools are imperfect. There are issues related to frequency response. Do we have a situation where the sensitivity of one of our devices, be it a radiation force balance or a hydrophone, has a different value if the frequency of ultrasound that it experiences changes? We could find that there could be issues relating to the orientation of the sound beam. And we'll look at examples of what are called hydrophone directivity and target incidence issues with radiation force balance. We may have issues related to limited spatial extent. And again, I shall be talking about a couple of examples of those. We may have environmental factors which are causing us to exhibit susceptibility. This could be electromagnetic, this could be other vibration issues. And finally, we may want to consider issues of dynamic range. Is our signal too small or too big to measure? And there's also the possibility, as I'm sure Sam Howard may be mentioning when he talks about therapeutic ultrasound, of the possibility of damaging our measurement equipment. Calibration is an important issue. Whilst it may be possible to set up a calibration facility within your own laboratory, often calibrations derive from the national measurement institutions, such as the National Physical Laboratory, the Physikalisches Technische Bundesanstalt in Germany, NIST in the States, NMIJ, and there are other NMIs around the world starting to set up calibration facilities. It's probably also worth considering what are the principal medical ultrasound uses. We've talked about diagnostic ultrasound, and therefore it's probably useful to be able to identify the sort of signals that we may get. So a not uncommon diagnostic pulse will be as seen here. These are typically short pulses. They often have a broad frequency range, a broad content of frequencies contained within there, and they can be medium to high pressures. Similarly, we may have therapeutic ultrasound. These are often much longer duration pulses. So in this case, we're looking at something that's about 170 microseconds long, but in practice, this could be all the way up to continuous signals. Because these tend to be mostly at one frequency, they have a very narrow frequency range. But there can be quite a wide range of pressures, from therapeutic ultrasound of physiotherapy nature, where we're looking often at quite low pressures, all the way up to high intensity focused ultrasound, where we could be looking at tens of megapascals pressure. So now let's start looking at some of those limitations, and we're going to start off by looking at the issue of frequency response. I'm going to start off by looking at a membrane hydrophone. And if we look at the frequency response of a membrane hydrophone, we can see that we've got a very broad response here. So we've got a resonant peak here at about 66 megahertz on this particular model, and we can see certainly in the range 0 to 40 megahertz we've got a very small amount of variation. So whilst it's going to be important to have this value quantified, because this value could move up and down depending on the sensitivity of the device, we will probably need to make a correction for the frequency dependent sensitivity, but that correction won't necessarily be huge. In contrast, if we look at one of the devices, a needle hydrophone, that we said was has a diffraction aspect or diffraction limited aspect to its frequency response, we can see here that if we look at this particular hydrophone, we've got quite a noticeable peak here and quite a noticeable trough here, and then a flatter region in the middle before eventually we start to tail off beyond about 32, 34 megahertz. We're starting to see a decrease in sensitivity. Again, for this particular model, different hydrophones have got a different response. Clearly, if we're making a measurement of a broadband pulse, particularly something that may be in this region here, centered about 10 megahertz, there could be quite a lot of variation as a function of frequency, and we would need to make sure we correct for that. And again, the international standards that are published have got methods well established as to how you might do that. Let's now look at directional response. Hydrophone output depends upon a number of factors. Firstly, we have the angle of incidence, we have frequency, and we have the size of the active element. And so what I'm showing here is a needle hydrophone that's actually at 90 degrees to the incoming ultrasound wave. And I'm showing here what will happen if we rotate that hydrophone around. You can see that the tip of the hydrophone doesn't move, it maintains at the center of rotation, but what we're doing is we're changing the angle of incidence. And I'm now going to show a video of exactly what happens to that experimentally. So the initial waveform as shown on screen is exactly with the hydrophone shown at 90 degrees to the waveform. As we start the animation to run through, we can see that the waveform becomes increasingly sharper and then goes back to its rounded shape once it goes back to 90 degrees in the other direction again. I'll rerun that animation. Now, the interesting thing is that in order to get very sharp signals, as we were seeing at the very top of the waveform here, we need lots more high-frequency harmonic content. In order to get this long, slow, sweeping peak negative going excursion, that tends to be dominated by our peak negative content, but also our low frequency content. So I will run that animation one more time, because we see that the peak negative doesn't vary massively as we rotate around, whereas the peak positive changes significantly. This is a fine example of how the hydrophone directivity function is affecting some frequencies more than others. We're also going to go on to talk about spatial averaging. Consider again a transducer that's radiating. And I'm going to look at a relatively large sensing element, which I'm going to sweep in front of that beam. We're going to start to record signal on our hydrophone when the right-hand edge encounters the beam of ultrasound coming out of the transducer. And it will stop when the left-hand edge leaves the beam of the transducer. So as you scan across the beam, there we go, we can see a nice broad profile of the transducer. I'm now going to look at exactly the same beam, but I'm going to look at it with a much smaller hydrophone. So clearly it's going to be much closer to the transducer before it starts picking up any signal. And this means that when we scan across the beam, we end up with a much narrower beam width, and we also tend to end up with a much higher signal as well, because we're averaging over a smaller area. So the important thing we need to look at this is that a large sensor has both underestimated the amplitude and overestimated the beam width. And without changing to a smaller sensor, we wouldn't have been able to tell this when we'd only made a measurement with our large sensor. Another thing that we need to look at is the issue of some of the environmental factors. Now, what I'm showing here is a not uncommon hydrophone response. I've been presented with some of these before. I happen to know that the hydrophone was at a distance away from the source that would correspond to a propagation delay of about 30 microseconds. So it was about 45 millimeters away. And yet we can clearly see in this situation that we're starting to record signal immediately. We have acoustic signal starting from time equals 0. Now, the only signal that gets to our hydrophone effectively instantaneously is an electromagnetic signal. And although we've got constant amplitude in this section of the waveform here, this is the response of the device to an electromagnetic interference. If we look in the middle section, we've got an interference here between an acoustic and electromagnetic signal. And it's only when we get to the end of the cycle, where actually our electromagnetic signal has died away, and we've got our acoustic only portion, that this small section of the waveform is the only bit which is getting us a correct measure of our acoustic amplitude. We also mentioned sensitivity and dynamic range. It's certainly true that bigger sensors provide a larger output. But as we've also seen, they can result in a more directional response, and they can result in a situation whereby you have greater amounts of spatial averaging. So choosing the size of sensor is careful, or something we need to consider carefully. If we have too small a signal, we can find that we have insufficient sensitivity, and we can end up with a noise-limited trace. And here is a classic example of a very noisy hydrophone trace. There are some things we can do in terms of temporal averaging which can improve that, and in fact that is actually the underlying trace that's beneath that noisy output. But we can also have the same situation where if we have a sensor that's too large, we can have too much sensitivity, and this can result in signal clipping. So if we look at this particular trace, whilst we've got nice clearly resolved peaks on the positive going excursion, we can see on the negative going excursion we're hard limited. We've clipped the signal and we're not accurately measuring what's going on in the peak negative. Thus far, I've only considered limitations on hydrophones, but it's also worth bearing in mind what can go wrong with a radiation force balance as well. Clearly, if we have environmental vibrations, we can see that we might get a transducer that's shaking around. Now, the principle of operation of a radiation force balance is that we're exerting a force down onto the target. Whenever this beam is arriving at a non-normal incidence, we can have a situation where we have a horizontal component of force. So not all of the force is exerted on the target, and therefore not all of that is recorded as a power. We may have a situation where our radiation force balance is too small, in which case we're not either accurately absorbing, or in the case of a reflecting target, accurately reflecting all of the signal that's incident upon it, which can lead to errors. We may also have a situation where our absorbing target is imperfect. And in this situation, we would get a reflection back towards the transducer. Certainly for some therapeutic transducers, they can be very sensitive to back pressure exerted on them. And so simply by the fact that there's a waveform reflected back to the transducer, this can impair the radiation output efficiency of the source device. So not only do we get an imperfect conversion of waveform that's going into the force that's exerted on the balance, but we're also impairing the output of the transducer. So to summarize the content of this webinar, I think it's important to realize that there are plenty of factors that can lead to inaccurate measurement. However, if we understand what can go wrong, we can help to ensure that it doesn't. And it's really important to make sure that our measurement devices are calibrated. I've provided you with examples of calibration curves for both the needle and membrane hydrophones, but similarly your radiation force balances need to be calibrated as well. So I conclude by saying that quantification of ultrasonic output is essential for regulatory compliance. It's needed in order to establish repeatable measurement conditions when we're undertaking trials. And whilst it's limited by the properties of measurement tools used, it can be accounted for with careful experimental setup and with sensor calibration. At this point, I thank you all for your attention, and I open the floor for questions.
Okay, well, thank you very much to Dr. Hurrell, and on behalf of the AIUM, our thanks to all of you who participated in today's webinar. Everyone, please remember to complete the post-test and the activity evaluation. We hope you enjoyed this presentation and will join us again for future webinars. So long, everyone.